So I'm going to talk to you about enzymes today. But first, I'd like to ask you a question. This is going to be really off topic here. Who remembers when this first came out? Yeah, October 2001. I know it's not the original one, but just go with it. The innovation that's really inspiring by this device, the iPod, I think you can really think about Steve Jobs as a major innovator. And if you were to poll about 10 of you in this room, I'd say 9% of you would say, yeah, Steve Jobs hits the top 5 on my list. And there's a reason why. He sees the potential, he saw the potential that the iPod could have in terms of bringing amount of storage that the MP3 player didn't have. Now you can listen to all 42 albums of, I think it's Michael Jackson, and if you really wanted to, you could probably put some Britney Spears in there too. I'm not going to admit to that one. But I really like what Steve Jobs had to say on the topic of innovation. And being that we're in the TMCI, innovation I think is really kind of pertinent. Innovation is the ability to see change as an opportunity, not a threat. I'd like to turn our attention towards a market, towards a topic probably more relevant to this audience, and that's synthetic biology. And first, I need to take your attention back to 1978, when the supply chain for insulin was very much under threat. Back in the day, the only way type 1 and type 2 diabetics could actually get their insulin was from the pancreases of pigs and cattle. And so Eli Lilly was quickly running out of that many animals to harvest. I think it was like 56 million for that year required to actually achieve that same amount of insulin they would need. And so a team of scientists in San Francisco came up with a really novel idea. And they used the simple technology that we use in the lab every day now, called recombinant DNA expression, which is simply just taking like molecular tweezers and grabbing the DNA that encode for the protein for the human insulin out of the pig and putting it into a microbe, a little tiny microbe that could be scaled up to an operation that allowed for sustainable production of this human insulin protein. And I think that's something remarkable about the idea of what innovation really looks like. If it's okay, I'm going to get a little personal, and this is really relevant to this topic of insulin. A few months ago, my mother was diagnosed with stage 2 pancreatic cancer. She, because of the surgery she underwent to remove the pancreas, she now is relying upon insulin, synthetic insulin, that's derived from the same technology that was developed back in 1978. And people, it's remarkable when you think about it, right? These team of scientists saw the threat to the supply chain and innovated, came up with a new option that couldn't be realized in that day and age. It just is remarkable. And so I just want to say innovation is the ability to see a threat as an opportunity. But I'd like to get to another story that's more relevant to this audience, and that is by asking a couple questions. What does water bottles, PVC pipes, antifreeze, and rubber tires all have in common? They all are derived from the same polymer, the same chemical building block. From a manufacturing standpoint, that's convenient. But what's not convenient is the process that's getting to harvest that chemical building block. It actually is responsible for 1/3 of the greenhouse gas emissions that in the world today, and that is simply just not feasible, simply not sustainable, relying on fossil fuels. And at Cemvita, we believe that we must do better. We have a moral obligation to find solutions, microbial solutions, fungi solutions, to solve some of these problems. And so I'm hoping that Kevin, who's in charge of our intellectual property at Cemvita, isn't going to shut my mic off here in a second. But let me tell you what Cemvita did. Because yes, we just patented it. So we took and looked inspired by nature. And it turns out nature is really, really adept at, as Marina just mentioned, at producing molecules that are biologically relevant and relevant to many different applications, such as, in this case, producing the same polymer that we would need, or chemical building block we need. So at Cemvita, we said, well, that's great. Really, we can use the same recombinant DNA technology that Genentech did back in 1978, grab the DNA that expresses or encodes for the protein, the enzyme, that makes this chemical, and put it into a microbe. But at Cemvita, we believe in taking it to the next step. We believe in going beyond just the bare minimum and doing the excellence. And that is when I look at a protein as a protein engineer, I see potential. I see what we can do with just the structure alone. And simply by changing one of the amino acids that's at the top, in the top letters, we can change its biological function or improve its biological function. And so in this case, we were able to double that titer of our chemical product. And in this way, we were able to create a green sustainable platform for producing our novel product, this product that normally would have just been taken so many years to develop. And now, before I can move forward about the scale-up operation, I'm going to let my colleague Edris Tahar tell you about that. But I hope you can see from my brief presentation here that really at Cemvita, we're doing something that hasn't really been done very well. And you can really see day by day how we're turning that dial away from the fossil fuel depletion and limited resources towards renewable enzymatic or microbial solutions. So with that, I'd like to say thank you.